1997, Professor Skoretsky won world fame when he discovered that most Jews of the priestly tribe are descendants of one man. In fact, he discovered the Kohen gene. Later, he showed us that 40% of Ashkenazi Jews are descendants of four founders' mothers who lived in Europe 1,000 years ago. And more recently, completed a study with surprising results on the origin and genetic characteristics of the Druze population in this area. Professor Skoretsky can rightly be regarded as the genetic archaeologist of the Jewish people, and he will tell us about the genomic history of the Jewish people. Thank you.
Everyone. As Dr. Ben Ishai mentioned, really, I'm a nephrologist, I'm a physician, and this was sort of a side hobby at the beginning, and it kind of became more than a hobby, and it began with the Kohanim, just like Dr. Ben Ishai mentioned, close to 15 years ago. And really, I'm a Star Trek fan. Many, I think, North Americans certainly were Trekkies like I was growing up. And this is Leonard Nimoy, as you'll recognize. He's not a Kohen, by the way. I know that on his website. But he was familiar with the priestly blessing, and you'll recognize that the Vulcan salute, for those who are Star Trek fans, is the priestly blessing, the way the hands are put out. So that's how it started, really, the interest. It began with a hypothesis. And I'll just give you the bottom line here. So, if it becomes a little boring during the talk, you can just remember this part, that genetic evidence overall indicates that all humans are part of one extended family. And then that same sort of genetic evidence, just fine-tuned, and one could do this with different populations, but if one applies it to the Jewish populations, that Jews scattered across most diaspora communities are members of a branch of this extended family originating in the Near East around 3,000 years ago. That's kind of the take-home message, and the rest is detail. So I think for those of you who want to detail, that's fine. But it's, as Dr. Ben Ishai mentioned, one can use many different tools, archaeology, linguistics, etc. DNA is just one other tool. There's nothing sacred or holy about it. It's an adjunctive tool that has to go together with others to be understood properly. But it is powerful, as we heard from Professor Chechanover. It has really changed our understanding of the biological sciences at the basic level, medical genetics, and then Dr. Ben Ishai used the word archaeology. And one can think of sort of two levels of using DNA in archaeology. One is anthropology, which looks way back at human origins. Where did human beings come from in the first place? Tens of thousands of years. And then genetic genealogy, which looks more at populations, communities, families, and we're going to focus on the second part. And I think for historical reasons, and Professor Chechanover gave a brilliant, as always, historical overview of a particular area. Recently, really, a historical scientific truth was corrected or brought to by Brenda Maddox in a very important book she wrote about everything that we're talking about is based on DNA, its structure, which we'll talk about in a minute very briefly, and Watson and Crick correctly, the late Crick, Watson, still get the credit, but their interpretation was based on an X-ray diffraction experiment done by this woman scientist from England, a Jewish woman, who, Rosalind Franklin. Now it's known, of course, what the whole story was there from Brenda Maddox's book, but it's, I think, always important to try to rectify that. Unfortunately, she succumbed at a young age before the Nobel Prize was distributed to probably, she had ovarian cancer, she was Ashkenazi origin, she may have had, we don't know, she may have had a predisposing genetic condition. And so I use that, and as a result of that, and Watson and Crick and so forth, we have the famous double helix structure, which when I was studying, this was taught in medical school. Now it's learned in grade school, and it's the double helix. And without going into detail, a set of billions of letters, but really the same four letters are used over and over again in different combinations to spell out what Professor Chechanover mentioned, ultimately the proteins. But one aspect is the fact that this double helix allows DNA to copy itself. So that's called DNA replication. Let's focus on that. If DNA replication were perfectly accurate with no errors like a Xerox machine, then it'd be great, be very accurate, but the world would be a boring world full of identical copies of the same sequence. But errors in DNA replication were put in or are part of the process, and that is what yields the richness of living and human diversity. But there is a price, and the price is occasional illness. Most of the diversity is not causing illness. It causes the beauty of diversity. And we have, therefore, we're at Rambam Medical Center and other medical centers, but there is illness, and we are enjoined there to heal, to try to heal, and that's what we're all about here. As I mentioned, most of these DNA sequence errors or replication errors, which can be called mutations, are silent. They don't make us look different, act different, or anything, but now, with modern technologies, one can see them directly, couldn't in the past. And they can be used as markers. These silent ones especially are very good to be used as markers because they're not influenced by selection and other factors, and one can analyze them. And these markers are the tools of the trade in population genetics when it's applied to trace ancestries, and also can be used to map real disease-causing mutations. So what are these markers? There's many different kinds. I'm going to mention two of them very briefly. One is called a SNP, single nucleotide polymorphism. It's simply a spelling error. So if we look at language, if the letter I goes to O, then you have a new word. If in the genetic alphabet a letter G goes to A, you also may get a new word if it's in a coding region, but you can certainly see that copying change. There's another kind of marker. That was a single letter change. That occurs very rarely. There's another kind. There are areas of the genome where you have repeated letters over and over again. And you may have 5 repeats, 6 repeats. In this room, at these spots in the genome, there's many different variations of how many repeats. And these are called simple tandem repeats, and they occur much more frequently. And a kind of an analogy is to think of a tree. And in fact, it's from the Bible, from Deuteronomy. Man is but the tree of the field. And these first kind, these SNPs, these single letters, you can think of as marking a new branch, a whole new branch. Something happened in history, a new letter entered, or there's a change. If that survived, the descendants of that individual, man or woman, will then transmit that and create a whole new branch of humanity which can be defined by that single letter change. And then you have these STRs, which are like twigs on that branch, and they tell us who belongs to one branch or another. And when you combine them, twigs on a given branch, using this tree analogy, is a lineage, a genealogic lineage. That's kind of the general idea. Now, where are these DNA markers? This is a schema kind of the chromosomes. They're located, obviously, on the chromosomes in the cells of our body. And here we see represented, let's say, the chromosomes of one man. It's a man because this represents 2 out of the many pairs of chromosomes with the different components to it. This part here is the Y chromosome. Just trying to free up the mouse here. Sorry. Because it's male. He's male. And mitochondrial DNA is another place that DNA is located. And the Y chromosome is an area which men transmit to their sons only. It's not present in women. And mitochondrial DNA, which is present in men and women, but only women can transmit it to their offspring. Men cannot. And therefore, Y chromosomes trace paternal lineages, and mitochondrial DNA trace maternal lineages. Now, that's the whole scientific background. Using that knowledge and an oral tradition, one can raise a hypothesis, and we tested the hypothesis, and basically, it's what Dr. Ben Ishai showed, that the markers demonstrate a limited number of paternal lineages shared among Jewish priests, many fewer than one would expect from a random group of men. We updated this recently, and the largest such paternal lineage, that is a twig on a branch, if we use the tree analogy, originates in around 3,200 years ago by analysis and calculation, and can be very readily recognized by a signature, which we called a modal haplotype, because it was the most common. Mode means the most common. And the idea here is really a simple idea. If I told my son, I think who's maybe in the audience here, a secret word and put it in a safe and lock the safe and told him, just tell your sons and tell them to tell their sons this word and tell no one else, never leak it to anyone, and then 1,000 years or 100 generations into the future or more, we opened the safe and took a group of men who knew the secret word, and just a random group of men, those who knew the secret word, if they kept it, should have similar Y chromosomes. That's a very simple idea, and that was tested. This is how rich the Y chromosome tree is. It's very rich. We have all men coalesce to a common Y chromosome, and then these are the lineages that we found for the priestly group, and this is updated as of 2009, actually, and this dominant one is 15 times more common among Kohanim compared to Israelites. we can use this then to hunt around the world for people who may have some affiliation. And we did that, and this is the first group we actually looked at in collaboration with a British group of scientists and Tudor Parfit, whose picture you'll see in a minute, who's going to be visiting Rambam in a couple of weeks, is the Lemba. And I just use it as an example to illustrate kind of an important point. And we look at, this is a picture of what I used to look like. This is someone from the Lemba tribe. And what is that? Are we related? Let's see. Lemba have a history that a man named Buba, many years ago, led a group out of ancient Judea to settle in what is now Yemen, the Hadramaut, and then years later migrated down the east coast of Africa, settled inward, and kept male, did not let other males in, but married local Bantu-speaking women. And therefore, their Y chromosomes should be very similar to the Y chromosomes one might see in the descendants of other Near East populations. And this is a picture here of Tudor Parfit from the School of Oriental and African Studies at the University of London. He frequents the area where the Lemba lived, which is northern South Africa, Zimbabwe, and Mozambique. And indeed, we found then, many years ago, at a low-level resolution, and now more recently at a very high level, that the top lineages that they have are similar to, identical to that of Kohanim in Ashkenazi, Sephardi, in Middle East and other origins, which we'll get to in a moment. So it's really quite remarkable. So this Buba story is not a Buba Maisa. So the Y chromosome signatures indicate that the Lemba and Kohanim share common Near East paternal ancestry. You couldn't have guessed this. And it's because of just different migration patterns from a common origin. Now, I use this, because I want to illustrate a point. I want to make a point, not just a simple curiosity, that this is a genealogic relatedness, and it relates to the work that was reported on this morning. And genealogic relatedness, it does not necessarily reflect genome-wide relatedness. It doesn't necessarily mean we share big swaths or stretches of the genome for medical purposes, let's say. And every genomic region has its own evolutionary history. And so that one can apply that to many different questions. So we studied at the level, let's say, of the Y chromosome, Ashkenazi Jews. There are many, the history of the origin of the Ashkenazi Jewish community. There's a conventional history, which is illustrated here, Roman exile, etc., the Rhine Valley, migration eastward, expansion, the Spanish expulsion, and there's always this question, how much entry is there from a Khazars, from a West Eurasian or other putative community? And in fact, the first person who raised this was Arthur Kessler in the book The 13th Tribe, and more recently a book by a professor at Tel Aviv University, The Invention of the Jewish People, etc. So we check that at all these levels, and at the Y chromosome it's very clear that Ashkenazi Jewish Y chromosomes are Near East. This pie chart just shows the branches, those branches that I talked to you about, being mostly Near East, and compared to European non-Jews, for example. There were two exceptions, Ashkenazi Levites and a very small number of Dutch Jews on the male side. I won't go into that in detail. And here we can see that for the Y chromosome, Ashkenazi Jews, their Y chromosome signatures are very much Near Eastern, similar to Syrians, etc., Palestinians, and so forth, and quite far away from Europeans and others. Well, we talked about the Y chromosome. The Y chromosome, half the people in the world do much better without a Y chromosome, and that's all the women. And if you ask them what the Y chromosome is good for, here's some of the votes that I've gotten, and especially with the Mondial, right? We have the Mondial now, and the question with sports, I'm not sure how many men are going to be working versus watching the Mondial, and probably women too, so it may not be exclusively. And here I am with some of our grandchildren. I just want to take a minute to say the fact that they're Israeli grandchildren is a credit to those who helped me and my family move to Israel, Professor Revach leading among them. And they're looking for their mother, not so much for their father, their grandfather, right? They're interested in the maternal lineage. So we looked at the mitochondrial DNA. It also has a beautifully rich tree, shown here, rooted in Africa and branching out beautifully. And the world populations are very sharply partitioned by the different branches defined by the maternal lineages, more sharply than any other part of the genome, Africa, etc., and so forth. We studied at the mitochondrial DNA level many different communities, Jewish and non-Jewish, and compared them. This is the story of the 4 founding women, which I won't go into in detail. Dr. Ben Ishai already mentioned it. Indeed, then we can see the Ashkenazi. Those parts of the genome tell us that really the Ashkenazi community began in the Middle East with maternal and paternal founding branches, with some entry and admixture from Europe. That subsequently expanded greatly over the history, 1,000-year history, leading to with what we won't go into detail, a higher incidence of certain Ashkenazi-specific transmitted diseases which are studied and so forth, including possibly what I spoke about with respect to the late Rosalind Franklin. We also have the historical event of, this is the edict of Ferdinand and Isabella of the expulsion from Spain, and there was both an expulsion and a forced conversion of Muslims and Jews to Catholicism. And if we look at the contemporary Iberian Peninsula population, and this is work done, led by Mark Jobling at Leicester. We had the privilege of being part of the work, but he led the work, and his group, published a couple of years ago. And look at Y chromosome signatures, for example, in contemporary residents of Spain and Portugal. We see the signatures that are very similar to the signatures that we see in Jewish communities for the Y chromosome and Muslim communities in the Y chromosome. Now, the big shift that was reported on is the shift from what I've talked about up till now, which is these uniparental, maternal, paternal only areas, to integrate across the whole genome, which adds a whole different dimension. This wasn't really technically very feasible until recently because of modern technologies has enabled this to be done, and therefore it has been done. And basically, the study that was reported on was an examination of 14 Jewish communities and 69 comparative non-Jewish communities from various parts of the world shown here. We haven't yet looked at the sort of what are called hidden or crypto-Jewish communities, but that's also being done. And that's this study that was released yesterday. I want to highlight the leaders of the study are Doron Behar. Doron Behar, I want to say a word. He's many of the physicians here know him. He's an internal medicine, he's a graduate of the Technion and specialized internal medicine, intensive care here at Rambam Medical Center. While he was doing his residency, at the same time, I had the privilege of supervising him in his PhD. He actually taught me more than I taught him. And he's gone on to do a great deal of population genetics work and is also a member of the Rambam staff. And he is now doing a postdoctoral fellowship with this Richard Willems, one of the world's leading population geneticists, at the Estonian Biocenter. I want to say a word about Estonia has decided, I don't know if it's a member of the OECD or not, but it has decided to invest millions in genomics and proteomics. And Richard Willems, a member of the Swedish Academy of Sciences, is leading that, and we have the privilege of working with him. Here are some other members of the lab who have been working on various aspects of population genetics. Gennadi's on this paper as well. Shai Tsur working on some other aspects. And the research was done by members of actually 8 countries. And just to show you, this is a representation of the conclusions. This is called a principal components analysis. I won't bore you with all of the details, but the Jewish cluster, this is the whole kind of all the communities, 14 Jewish, 69 non-Jewish, color-coded, etc. And the Jewish communities cluster very tightly. And who else is in this cluster? The Levant populations, Syrians, Lebanese, Palestinians, etc. That's who we overlap with genome-wide, not Y chromosome or mitochondrial DNA, genome-wide. So we resolved the Middle East substructure much more carefully than been done before and showed the Jews from communities covering around 90% of the contemporary population cluster with current Levant populations. Those are the populations I just mentioned. And we confirm this with other approaches, and maybe I won't, this is many different analytic approaches were used. This shows the cluster again. At the same time, very recently, and this is very nice when this happens in science because it doesn't happen that often, because once a study's published, there isn't a great incentive for scientists to replicate it because it's hard to publish again. But here, totally independently, another whole group, here's the authors, mostly led from the United States, also a geneticist from Tel HaShomer, did a very similar study, came to a very similar conclusion. This is a different representation. I just want to highlight one aspect of their study, which is of historical interest. This was published in the American Journal of Human Genetics. Same within a week of each other. These two studies are published in parallel with we knew it was going on, but certainly completely independently. Look at this major sharing between Iranian and Iraqi Jews. So the interpretation here, and it goes back 2,500 years. What's the interpretation? That this is the Babylonian exile. This is the current fingerprint of the Babylonian exile. That community then thrived, as we know, in what is today Iran and Iraq. They've since left, of course. And we see that signature. So we studied many communities. This is the island of Jerba. Just some pictures to show you, the interesting communities. Many of you have visited. Mostly I get the DNA. Other people visit. And the three major communities in the Indian subcontinent. Some pictures from the Bnei Israel community. We have one of our most distinguished physicians here from this community. The Cochin, Bnei Menashe in northeast India. Now guess what community this is. We haven't checked their DNA yet. Anyone want to guess? This is from an archival record. You look up here and you can guess, and Professor Shechanover is my main partner in trying to further this work. It's the Kaifeng Jewish community, and now it's going to be easier to study them because some members have arrived here and are in Israel now. Here they are. There's actually even a bigger group, and certainly they have a strong tradition. Now, if we say there's this overlap, one can apply these same principles to many other populations, and we did the study that Dr. Ben Ishai mentioned on our Druze neighbors and our and residents here, both in Israel and in Syria and Lebanon. Don't ask how we got those samples from Syria and Lebanon, but we did get some. And basically, this is the demography of the Druze population in Syria, Lebanon, the Carmel, the Galil, the Golan, a little bit in Jordan. And what we found is something that really surprised me, at least. We see in a tiny, tiny area, let's say here in the Carmel, you see the names of the villages. Some of you will recognize them. And these are the color codes of the various different lineages, let's say of the mitochondrial DNA, but the same is true of the Y chromosome. And there are many different lineages among the Druze. And the religion was declared only 1,000 years ago. So how could this be that in a small, tight community, geographically confined, and with a 1,000-year declared history, well, the secret Druze religion says there were Druze antedating the actual declaration of the religion. And we, in fact, our only sort of inescapable conclusion at this point is that the Druze population here, a representative snapshot of the population genetic structure of the Near East, like they're a reference for the genetic structure of the Near East in antiquity, where global diversity evolved. When humans moved out of Africa, this was one of their main stations where diversity emerged, and we can see that among the Druze population. So it becomes a reference and also important in disease mapping. I want to finish with another population, and that is African Americans. Now you might say, why talk about African Americans in this context? And I'll try to put it together. First of all, I mentioned I'm a nephrologist, so medical questions are really the driving question. Kidney disease is a very important disease, and we would like to prevent it. Professor Chikandar mentioned prevention. So we have to understand why people get kidney disease, why they end up on dialysis. Well, one clue is there's a disproportionate prevalence of patients of African ancestry who develop kidney failure, except for an exception. The African ancestry of African Americans is West African. We noticed and published, and again, Dr. Behar and Liran Shlush and others here at Rambam and the Professor Pollack's AIDS clinic and service, we collaborate closely on this, note that Ethiopian Jews have a low risk for kidney failure, very low risk. What protects them? Now, we could check non-Africans with African Americans, but we know that genealogically and evolutionarily, it's better to compare populations which are closer. So we did this, and we found certain risk markers. We, as a whole big group, so I won't name them all. You'll see a picture perhaps later. Shai Tzur, who you saw earlier, was one of them, and others. And we found risk markers that really sharply demarcate sub-Saharan Africa from Northeast Africa, and some of these are completely absent in Ethiopia. So we say, oh, this must be the area of the genome where the risk or protection exists. So then we can do this kind of mapping and find where in evolutionary history of the genome this mutation occurred which causes West Africans to have a higher prevalence or tendency to kidney disease. We've done that, and hopefully, I think we may have found possibly the cause of the mutation. It's now under review, so I won't talk about it very much. And this gets right back to personalized medicine and health as beautifully elucidated and enunciated by, so I won't go over it. But again, here we used, I'm going back to this example. This was just a interesting, I think quite interesting barcode, surprising, of relatedness genealogically at the Y chromosome level. But one can do that for, instead of saying, do we share a common father? Do we share the same risk for disease? Do we share the same predicted response to a drug or not? Use the same principles exactly. So when I use this example, I finally changed my car, but had this for about 13 years. And if I went to the mechanic and said, I have a problem with my car, and I said, well, my car is green, that wouldn't help him. If I told him the part with this serial number is the problem, then he can really fix the car, okay? So that is the whole issue. And this is a lifelong process. Professor Chikana mentioned the epigenome and the complexities. We're not stuck with just the genome we're born with. It changes over time. It's dynamic, and therefore we change over time. We age over time. And by having a biobank, and Professor Chikana has led a very important initiative for the state of Israel to have an Israeli-wide biobank, which is going to be a it's more precious than oil in terms of trying to help further humanity. Now, I've talked about genetics a lot. I borrowed this cartoon from way back when, more than 10 years ago. I saw it and I kind of liked it. Geneticists sometimes get a little carried away, and they think, we find the gene for everything, including making us think that everything is genetic. And it isn't. And when we see diseases like kidney disease, etc., we have a genetic endowment, but we're not slaves to it because there's environmental, dietary, and other modifiers. And the Talmud already knew this and predicted it without knowing about DNA. The Talmudic scholars enjoined us to be a student of Aaron. Aaron, what qualities did he have? He loved peace, pursued peace. You don't have to be a direct descendant of Aaron to have these attributes. You can learn them. I mentioned some of the people working particularly on population genetics. This is Gennady, Shai, etc. As Dr. Ben Ishai mentioned, we work on many other topics. And, we're mostly in the lab. I don't want you to get the wrong impression, but we try to have fun sometimes. And here's the lab team in the Hula Valley, and these are people working on other aspects, telomeres, stem cells, etc. I won't name them all, and so forth. So we also try to enjoy what we're doing as well and enjoy the country. And I thank you very much, and I'm pleased to be here.